Good morning, students. Today we shall discuss a bit of the nervous system, which is reticular activating system, electroencephalogram, and sleep. These three things are interrelated, and that's why they will be discussed simultaneously. Now, first of all, we shall talk about the reticular activating system, or in general, reticular formation. Now, what is reticular formation? Reticular formation is an extensive network of neurons which are present in the brainstem, that is the midbrain, pons and medulla, subserving multiple functions so far as the ascending systems are concerned as well as the descending systems are concerned, which passes through the brainstem, maintaining a communication between the spinal cord and the cerebral hemisphere. So if this be the fact, we must have a clear-cut idea regarding the reticular formation. Now, as I told you, it's an extensive network of neurons or nerve fibers communicating with each other, forming multi-synaptic pathway, and they are divided into two major portions. One is the lateral division of reticular formation, another is the medial division of the reticular formation. Now, if we consider the lateral division, then it contains various important vital centers of our body, namely the cardiovascular system, the respiratory system, the vomiting center, then the sneezing center, coughing center, all these things, they are the different vital centers located in the lateral portion of the reticular formation in the brainstem. But so far as the medial part is concerned, that is very, very important. Now the medial part is once again divided into two systems. One is the ascending reticular system, another is the descending reticular system. Now so far as the descending part is concerned, they maintain the muscle tone by making direct communication with the spinal cord as well as modulate different sensations which are entering through the dorsal horn of the spinal cord. That is about the descending part of the reticular formation. Now regarding the ascending part of the reticular formation, which is our topic of discussion today, and that is also known as the ascending reticular system, or also known as the reticular activating system, or RAS. So what is RAS? Now reticular activating system, or RAS, is an ascending pathway, polysynaptic in nature, present in the midbrain, pons and medulla, that is in the brainstem, occupying the core portion of the brainstem, that is this thick arrow core portion of the brainstem, and is going to the thalamus to communicate with the different thalamic nuclei. And from there, the fibers, that means from the thalamic nuclei, fibers project into the different parts of the cerebral cortex. Different parts of the cerebral cortex, virtually all parts of the cerebral cortex, they project from the thalamus. This portion, that is the part of the reticular activating system from the thalamic nuclei to the different portions of the cortex, is known as the non-specific thalamocortical radiation. These fibers, these arrows, which are going to the extensive parts of the cerebral cortex. Now here, if we come into the brainstem portion once again, we are seeing that these reticular activating system, or RAS, occupying the central core of the brainstem, is getting communications by collateral fibers from the specific ascending tracts. This is the specific ascending tracts. Specific ascending tracts. They are giving collaterals to the reticular activating system. This anatomical communication is very, very important. I will tell you later. But they give communications or collaterals to the reticular activating system. So, what is RAS? Multisynaptic pathway present in the brainstem, getting communications from the specific ascending tracts, and after relaying into the thalamic nuclei, they are going into every nook and corner of the cortex to keep the cortex in a steady state. So that is about the anatomical part or functional anatomy of the reticular activating system. Now, the functions of reticular activating system or RAS. What are the functions? They subserve four important functions. Number one, they are responsible for the maintenance of the consciousness of the subject or of the cortex. Second one is the alertness of the cortex or of the subject, of course. The third one is the responsiveness of the different stimuli perceived by the cortex, and the fourth one is the wakefulness of the cortex or of the subject. So, what are the different functions subserved by the reticular activating system, which is also known as the non-specific sensory pathway passing through the brainstem? It subserves four functions, that is the consciousness. Number two is the alertness. Number three is the responsiveness. Number four is the wakefulness of the cortex. So that is the main function of the reticular activating system. Now the question, what is the necessity of maintaining all these four functions? Because to do something with proper precision, for the perception of different sensory modalities, the cortex must be conscious, the cortex must be alert, it must be wakeful, and it must be responsive to numerous stimuli coming from peripheral parts of our body to reach the perception area of the cerebral cortex. So that is the main function of the reticular activating system. Now we will go to the applied aspects of the reticular activating system. Now, if the reticular activating system is blocked in any portion of its extent in the brainstem, then what will happen? Then the four functions as already mentioned will not be there or will be suppressed. That is, the subject will lose his or her wakefulness, consciousness in that sense, alertness and responsiveness. So the cortex will be suppressed as a whole and the person will fall asleep or will feel sleepy. If the reticular activating system is interrupted by anything, say for example a tumor in the brainstem, suppressing or compressing the transmission of the impulse through the non-specific reticular activating system. Or the second applied importance is this, that whenever the sedative drugs or the hypnotic drugs or different anesthetic agents are given, they act by causing interruption of the synaptic transmission in the reticular activating system. Now perhaps you know that these reticular activating system are having two important pathways so far as the neurotransmitters are concerned. One is the noradrenergic pathway, another is the serotonergic pathway. Noradrenergic pathway and the serotonergic pathway. Now these hypnotics, sedatives or the anesthetic agents, they cause interruption of this noradrenergic and the serotonergic pathway. So the transmission of non-specific impulse through the reticular activating system is blocked and the effect of the anesthetic agents or the sedative agent or the hypnotic agents are manifested. So that is about the drugs which act at the level of the reticular activating system. So that's all about the reticular activating system. Now we will switch over our discussion to EEG or electroencephalogram. Now, if you know or if you have something conceptually in your mind regarding the electrocardiogram, where we recorded the electrical activity of the heart by placing numerous electrodes in different locations, strategic locations of our body, so that virtually all the electrical activities of the heart are recorded in a graph paper, and from that particular paper we can interpret whether there is any abnormality in the cardiac impulse transmission or cardiac rhythm or any abnormality so far as the electrical activity of the heart is concerned. Similarly, electroencephalogram is the recording of the electrical activity of the brain by placing numerous electrodes on the scalp. The strategic locations of the electrodes, I am not going into the details because that part is concerned with the recording of the EEG, which you will be able to find in the neurology department where they are recording it regularly. Now, these electrodes are placed in the scalp in such a manner that virtually all the portions of the scalp is covered. That is the frontal region, occipital region, and the two temporal regions, and in between numerous 10 to 20 electrodes are applied, so that basically virtually all the electrical activities of the cortical cells of the cerebral cortex are recorded or can be cached. So this particular recording, which will be recorded in a graphical paper like that of the ECG paper, is known as the electroencephalogram. The process is known as electroencephalography, and the instrument is known as the electroencephalograph. Now let us come to the different waves of the EEG. The most prominent wave of the EEG is known as the alpha wave or alpha rhythm. Now, for the production of the rhythm alpha or alpha rhythm, there must be some conditions which should be fulfilled. And these conditions are: the person must be awake, that means not sleeping, but the eyes are closed, and the mind is wandering, that means the mind is not focused on a particular problem or particular thinking, known as wandering minds. If these are the conditions, then the waves produced in EEG is known as the alpha wave or alpha rhythm, having some frequency, characteristic frequency, and the amplitude. The normal characteristic frequency of the alpha rhythm is 7 to 14 hertz frequency, and the amplitude is up to 50 microvolt. Up to 50 microvolt. So the amplitude is up to 50 microvolt and the frequency is 7 to 14 hertz. These are the characteristic features of the alpha rhythm or alpha wave, and it is the most prominent wave in EEG, recorded from a normal adult person, particularly from the occipital and the parietal region. Occipital and the parietal region in a normal adult subject. So that is about the alpha rhythm. Now, if this alpha rhythm is interrupted by some external stimuli, then the configuration of the alpha rhythm changes altogether. What happens? Now, if the person suddenly opens his or her eyes, or if he or she starts thinking or focuses on a particular problem, then what happens? Then this alpha rhythm is disrupted. There is desynchronization of the alpha rhythm, leading to formation of a wave whose frequency is very high and the amplitude is very low. So that particular rhythm is known as the alpha block rhythm, because the alpha wave, normal alpha wave, has been blocked by some changes in the external stimuli. Eyes are opening, mind is not wandering, focusing on a particular problem, or there are some stimuli which are given to the subject, say for example bright light is focused on the eye, or the person is touched, or the person is given pain sensation, then this alpha block rhythm or desynchronization rhythm will appear in the graph paper. That is alpha block. The next one is the beta rhythm or beta wave, which is having a very high frequency, 15 to 30 hertz frequency, and is a very low amplitude, 15 to 30 hertz, and is a very low amplitude. So it will look like this. So that is the characteristics beta wave, which is normally found in adult person from the frontal region. That is, the electrodes placed in the frontal region will record normal adult subject from a normal adult subject, the beta rhythm, which is having a very high frequency of 15 to 30 hertz and a very low amplitude. Now, the next two waves, they are normally not recorded in an awake person. These two waves, that is the theta wave as well as the delta wave, they are known as the slow wave rhythm. Slow wave rhythm, that is, their frequency is very low, but the amplitude is very high, like this. I'm talking both about the theta and the delta, particularly the delta wave, which is a characteristic wave when the person is asleep. This is known as the slow wave rhythm, whose frequency is very low, only 1 to 3 hertz. I'm talking about the delta, 1 to 3 hertz, and the amplitude may go as high as more than 100 microvolt. So that is a very high amplitude wave, but the very slow wave. Quite expectantly, these waves are supposed to be found when the cortex is suppressed. That means during the sleeping state. So that is about the different waves of the EEG. Now we will come to the factors influencing EEG. What are the factors which will influence EEG? Number one, the factor which influences EEG is the hypothermia. Whenever the body temperature is diminished remarkably, then the amplitude of the waves are reduced remarkably. Hypoglycemia, when the blood sugar level falls, there will be also low amplitude waves. Diminished blood level of cortisol hormone, known as hypocortisolemia. In that condition, there will be decreased amplitude of the waves. That is, the configuration of the waves are getting changed due to these conditions. Hypothermia, hypoglycemia, and then the hypocortisolemia, and most important, very, very important, the hypocapnia. That is the only exceptional situation where the amplitude of the wave is increased. So if the PCO2 level of the blood is diminished, the condition is known as hypocapnia, and during hypocapnia there will be increased amplitude of the EEG wave. And that is the reason that whenever a person suffering from epilepsy, I will tell you later on, is not being able to be produced the particular changes in the epilepsy, he or she is asked to hyperventilate so that the carbon dioxide is washed out. There is hypocapnia, and the hidden waves are manifested as high amplitude waves. So these are the four conditions where the EEG waves are changed or altered. Now we will come to the next part. The next part is how the waves are generated. What is the mechanism of genesis of EEG waves? Very, very important to know that. We shall have to come back to the RAS portion once again. We call it that whenever the EEG recordings are there, it is nothing but the algebraic summation of the local graded potential of the dendrites of the superficial cortical cells. There are important conditions which are there. to register the thing as EEG recording, it must be the dendrites of the cortical cells whose local potentials, when are algebraically added, they give a picture which is known as the electroencephalogram. Right? So it is a local graded potential which upon addition algebraically gives the configuration of the EEG. So that thing which we record from the surface of the scalp. Now, if you go into this diagram once again, that is the reticular activating system. So what is happening? We are seeing that the non-specific thalamocortical radiations are making synaptic connections with the superficial dendritic cells of the cortex. This is very, very important. That's why I have shown this in this diagram, that the non-specific thalamocortical radiation, which is a part and parcel of the reticular activating system from the thalamic nuclei, are going to make extensive synaptic connections with the dendrites of the superficial cortical cells. So, for the genesis of the local potential of the superficial cortical cells, quite naturally, anatomically as well as functionally, this RAS is playing an important role. So, whenever there will be any alteration in the activity of the RAS, so there will be definitely alteration in the activity of the local graded potential of the dendrites of the superficial cortical cells, and thereby obviously into the EEG recording. Do you get my point? So that is very, very important, and that's the reason that whenever the RAS is disturbed by any superficial impulse or other external stimuli, that is the touch, pain, temperature, by virtue of this collateral connection, you just see the collateral connections, they are being transmitted through the specific ascending tracts, and as they are related collaterally with the reticular activating system, they are stimulating the reticular activating system, and that's the reason whenever the touch is given to the subject, whenever the painful stimuli is given to the subject, they are stimulating the reticular activating system, and by virtue of this non-specific thalamocortical radiation, they are influencing the potential of the superficial cortical cells whose dendritic potentials are recorded as the EEG. So this anatomical connection is obvious, which can influence the EEG waves. So that is the mechanism of genesis of EEG waves. Now, lastly, we will come to the applied aspects of the EEG. Now, EEG from time immemorial is a very, very important mechanism by which a number of diseases of the brain are diagnosed, of which the most important is the epilepsy. Now, you know, epilepsy is a condition where there is abnormal discharge from the cortical cells, and as a result of those abnormal discharge, there is some changes in the EEG waves which can be detected by the neurologists, right? The epileptic waves, different types of epilepsies are there: grand mal epilepsy, petit mal epilepsy, absent seizure epilepsy, whatever may be the type of epilepsy, those can be diagnosed by EEG during the attack. But in between the attacks, if you try to record the EEG, that will initially show a normal value. For that, you shall have to manipulate the subject by giving photic stimulation or powerful light stimulation to the eye, or as I told you earlier, you shall have to ask the subject to hyperventilate so that the hypocapnia is created and the hidden EEG waves characteristics of epilepsy will be manifested. So that is the most important use of EEG. Apart from the fact that EEG can also localize the location of a tumor in the brain. Location of a tumor in the brain. But nowadays the CT scan is used instead of EEG, and the CT scan can precisely locate the nature of the tumor as well as the location of the tumor. But still EEG remains the cornerstone of the diagnosis of epilepsy, and they are the first tool to the neurologist for diagnosis of epilepsy or epilepsy related diseases. So, to summarize, the reticular activating system, which is a system of multisynaptic pathway stimulating the cerebral cortex, and the cerebral cortex remains awake and wakeful so that it can perceive the different sensation precisely and locate the sensations. And as a result of the activity of the reticular activating system or RAS, there is production of electroencephalogram, and those electroencephalograms show a number of different waves, and by studying the waves we can arrive at a conclusion whether the waves are normal or there is some backward abnormal electrical activities of the brain. So that's all for today. We will go to the sleep section in my next class. Thank you.